All right, so bacterial cells, if they undergo typical binary fission, okay, and one cell divides into two cells, so binary fission, those two cells should be identical to each other. We talked about mutation, so if there was some mutation that remained when DNA was replicated and then went into the new cells, it's possible this way that the organisms can pick up some amount of genetic diversity. So that's what we want to get into now, is genetic diversity in bacteria. So when other organisms are going to reproduce, they undergo meiosis, and meiosis produces gametes that are unique. They're completely unique, and when then they fuse together, they have all unique offspring. So thousands and thousands of different combinations, hundreds of thousands of different combinations because of genetic recombination. So some bits of one chromosome swap with another chromosome. So you can get maternal and paternal DNA swapping and then being part of the same chromosome that then gets passed on to your offspring. So essentially, you have chromosomes from your mother, you have chromosomes from your father, but when you produce gametes, you can pass on chromosomes to your offspring that are not just 100% from your mother, they're actually your mother's chromosomes that you inherited, and a little bit of your father's are spliced together into a new version that then you pass on. So all this unlimited possibility. Bacteria typically don't see that, so how can they be different? How can they adapt? How can they change? How can they evolve? So mutation is one pathway. The other is horizontal gene transfer. So that's what we're going to focus on now. There are three main types of horizontal gene transfer that we're going to look at. Right now I'm going to go over transformation and conjugation on the first two. And then I'll have a second lecture video on the process of transduction. Okay. So transformation. If we have a bacterial cell, and in that cell we have its DNA. Probably should have drawn this a little bit bigger, but that's its chromosome. In the environment are bits of DNA from other organisms. It's called exogenous DNA. So it's DNA that's outside the cells, that's in the environment. This is from other cells that have died. Dead cells, get broken down and their DNA is released. Now, not all cells can pick up that DNA, and in fact, it could be dangerous to many cells. Some cells produce DNA enzymes that they release that can destroy these bits of DNA, so they can't be taken up, which can cause then mutation and changes that are unwanted in the cells. But some can. All right. So some bacterial cells are called what we call competent. So competent cells, I'll draw this a little bit bigger, have embedded in the cell, so maybe it's the cell membrane, we'll kind of expand this out a little bit here, in the cell wall. We have some DNA binding proteins. And now this exogenous DNA from the environment can actually be taken up, bound to these DNA binding proteins that are part of the cell wall and then pulled into the cell. So now you have the chromosome in the cell and a bit of foreign DNA the foreign DNA is brought into the cell through these binding proteins. We can then get genetic recombination. So what will happen is then if the cell wants to do this or has the enzymes that are capable, or we should really say, to do this, then it will actually splice a bit of its DNA, cutting it open, and then it'll insert. So an insertion of the foreign DNA into its own chromosome. Right? And so now we have additional genes from other bacteria. They could be genes that allow it to use their nutrients that it couldn't use before. They could contain antibiotic resistance genes, a variety of different things. they might not be able to do anything particular useful for the organism, right? And just provide some extra bits of DNA in there. So it's not specifically choosing to do this for a specific reason. It's just something that would happen to the cell, and it happening to many cells over time. Some cells would pick up information that's useful or helpful. Those cells would then survive. Those cells would then grow more, divide more, and then pass on those genes to other cells, which could then potentially take up more genes from the environment and add and add and add to the diversity. So one way that bacteria become more genetically diverse is essentially taking up DNA from the environment. So let's take up DNA from the environment, and we call that transformation. Not all cells can do this. Only cells we said that are called competent cells can do this, that have these binding proteins. Now, if in a laboratory situation, someone wants to add specific DNA and incorporate it into a bacterial cell, they can force it in even if the bacteria is not competent. So there's a technique called electroporation. So electroporation can, using electrical current, open up bits of the cell wall and cell membrane without damaging the cell, allowing then bits of DNA to then travel across and enter into the cell. All right, so that's a laboratory technique. It doesn't happen in nature. In nature, either the cells are competent to do this, or they're not competent to do it and they can't do it. Okay? So, and that's all we're really going to go into, not a lot of extra detail. So know the definition of it, and just the basic idea here. All right, so the next thing we're going to do is look at conjugation, and that's the one that's going to take some more time. Okay, so we got that erased pretty quick here. So conjugation is the process that we're going to spend quite a bit of time on. And then transduction also. And transduction will carry over also to your fourth exam as well. And we talk about virology. So conjugation is where we have one bacteria that has its own genome, obviously. And then in addition to that, it has plasmid DNA, which we've talked about already before. This particular type of plasmid is what we call an F-plasmid. The F-plasmid has genes within it to build a pilus, for the construction of a pilus. Okay. So if you remember, the pilus is an extension that a cell can make from its cell membrane and cell wall outward that it can use for attachment. And potentially some cells can use it for motility as well. In this particular case, a sex pilus, right, is a unique type of pilus that's going to connect one bacterial cell to another. So we will get different colors. We see another cell here. Here's a bacterial cell. Here's its chromosome. And it doesn't have the F-plasmid at all. So cells with an F-plasmid are going to be called F+ cells. And cells without the F-plasmid are going to be called F- cells. Right? F+, F-. The information contained in this cell will then allow it to build this pilus. So we'll kind of move it here as a connection. And then join the cytoplasm, of both cells. All right, what's going to occur, and I'm actually going to swap the position of the chromosome and the plasmid here, just to make it a little bit easier for us to see what's going on. So I'll relabel this here, so it's not going to be the plasmid. The plasmid's going to be here. And the plasmid, for what I'm going to do here, I'm not going to draw it. Usually I draw these kind of with like I did a little squiggle that kind of indicates that this structure is DNA and it's a helical structure, like this. But for the purpose of this, to make it a little more simple to see, the plasmid, I'm not going to do the little helical structure. I'm just going to draw two rings like this. It doesn't actually look like that, but it makes it a little easier to understand what's going to happen here. There is a place on the plasmid where it will be cut. Okay, so we'll take this and we'll cut the one single strand of the DNA, not both strands, just a single strand. And then what's going to happen is this piece of DNA, the one single strand of DNA, is going to be pulled off of the other strand, right, and separated from it. As that happens, DNA replication will occur and a new strand will be built along this existing template. Right? And then this process will continue and you'll kind of pull away more and more, and then you'll build more and more DNA like this, and this piece here will get longer. Eventually what's going to happen, I probably should have drawn this bigger to start with as well, so I'm kind of figuring this out as we go along. Again, just so you can see better, I'm going to make this a little bit larger, or else you're not going to really see what's going on. This would be a very fine structure. It's not as massive as I'm drawing it here, but in order to see what's happening, I have to really expand it. So this piece of DNA is going to be growing. New DNA is being made in this direction. This piece of DNA, this is single-stranded DNA, is being pulled away from the other strand in the plasmid, and it's being fed through the pilus. So it's moving from the F+ cell to the F- cell. Eventually what will happen is the process will complete and end. And here you'll get, new DNA built. This will be the double-stranded DNA. It'll go back to double-stranded DNA. Here, the single-stranded DNA will then move into this cell. As it moves into this cell, what's going to happen? DNA replication will begin, and it will actually start building the other strand. All right, eventually this process will go ahead and complete as well. They'll just join like this. And then this cell will become an F+ cell, because now it will have the entire F plasmid with the same information that it can pass on to another cell. The pilus itself will break down or be pulled back, into the other cell. Use this guy here to erase. And the membranes and cell walls will kind of go back together again. Like that. And that's it. So essentially what's happened here in this type of conjugation is, an F+ cell passes single-stranded DNA to an F- cell. Get rid of this so it's on the way. All right, that's the basic idea here. So again, this is plasmid. So it is extra-chromosomal DNA. There could be many different genes, inside the plasmid that are then passed on to the other cell. And that way this cell now gains new genetic information. When it divides, the plasmid will be replicated as well, and it would get passed on to the new cells through binary fission and so on. But as an F+ cell, it could then find other F- cells, and then it can pass on the plasmid to those cells, and so on and so forth. And so the information can just be passed from cell to cell this way. Right? And so cells that don't have the information will gain this new bit of information through this extra chromosomal DNA called the plasmid. All right, so that's the first type of conjugation, which involves F plus and F minus cells. Now, so that's this first part here. The second part of looking at conjugation is where we have cells that we call HFR cells. All right, and so this is going to be a little bit different, similar, right, but a little bit different. So I think it's going to be easier probably if I erase this. I'm going to erase this one. I'll try to keep these cells so I can not erase everything. And I'll leave some of the terms up here. This is still going to be the F minus cell. This is no longer going to be the F plus cell. This is going to be our, I'll rewrite this if we need to add stuff about the pilus here. There's going to be another pilus. This is now going to be what we call an HFR cell. So HFR stands for high frequency of recombination. All right, so recombination, that's what happens in our chromosomes when we make the gametes, when a bit of one chromosome then overlaps with another chromosome and they swap genes, and then you pick up genetic information in your actual chromosomes. So that's what's going to happen here now to this F minus cell. It's not going to receive a plasmid, right? There isn't a plasmid here. So there's not going to be any extra chromosomal DNA in this particular case. The genes here are not separate. With an HFR cell, what you have is the F-plasmid genes have actually been incorporated directly into the DNA. So the pilus forming genes and others are now incorporated into the bacterial chromosome. So this is the bacterial chromosome. And then you have the additional genes that have been incorporated into it. So that's something different. So now what's going to occur is just like before, when these genes are expressed, they're going to control the production of a pilus. So they'll find an HFR cell will find F minus cells. So F minus cells are the ones that have none of these genes. So whether they're going to be attached by a pilus to an F plus cell or an HFR cell, they're the ones without. They just have their normal bacterial genes in the chromosome. So a pilus forms again here. So it's from this particular cell. And now what's going to occur is we're going to cut, all right? Well, again, one strand here of this DNA. Probably have to do it like this here. And pull it off. And what we're going to do is actually start to feed often this chromosomal DNA into the pilus. So you can see the pink here is supposed to represent the F-plasmid genes, the ones that have been incorporated into the chromosome. Kind of do this like this. So we got this strand here. This strand has been pulled away. Hopefully you can kind of see that. And so this is actually a bacterial chromosome. It's not the F-plasmid genes. So although the F-plasmid initiates the process and controls the production of the pilus, they're not necessarily being passed to the other cell. What will happen is this process will continue as it did before, and you'll pass this new piece of DNA in here. typically the process stops before it either reaches or completes the F-plasmid genes. So you may get a small portion of those genes, but not the entire thing. Now, in this particular case, what will happen is in the new cell, okay, I'll do this, you'll get DNA replication, and you'll have this double-stranded DNA. So now the double-stranded DNA, I'm going to redraw this, lining up like this. This double-stranded DNA will then match up with the host cell's chromosome. And then again, if it has the proper enzymes, it will then cut here, splice that information into the chromosome, like that. So it's been incorporated into the bacterial chromosome. So this does not become an HFR cell or an F+ cell. This remains an F- cell, because it typically will not receive the complete set of genes required to produce a sex pilus to pass information on to another cell. What it will receive are new bacterial genes. So that is a recombination. It's a new combination of genes that it has received from another organism, and now its genome has been altered or changed from this process. Okay, so it's still through conjugation. Conjugation involves a pilus. It involves one bacteria attaching to another bacteria, passing DNA through the structure to the new cell. And the new cell gets that genetic information, which it can express, and then potentially, help that cell be useful in some way, or not. Maybe it doesn't do anything in particular for the cell, but it's adding to the genetic diversity of that cell, which could be selected for based on nutrients that it can use or resistance to stress in the environment and the other sorts of things. So the two processes of conjugation involve the separate circular plasmid that's not part of the chromosome. That's just called the F+ cell, or HFR cells, which have those genes incorporated into the actual bacterial chromosome. When it initiates the pilus, those genes typically don't pass through, but some of the genes of the bacteria do. So they get passed across. Again, I didn't draw this in here, but replication, would occur here, and this would stay a complete genome there. But a piece of this would get passed on to the other cell, and then that piece would then get a second strand to become double-stranded, and then it would potentially be incorporated directly into the chromosome. So the chromosome of a bacteria, the recipient, so the F1 can also be called the recipient cell, will change, and it will get new genetic information. All right, so those are the first two mechanisms of bacteria becoming more diverse genetically. Transformation is where bits of DNA are taken up from the environment. Okay, so just kind of random pieces of DNA are brought into the cell if the cell was competent, we said, which means it has proteins that can bind the DNA and actually bring it in. Not all cells can do that or would want to do that, but because it could cause problems really for the cell. But some cells do. Conjugation is a more specific process where bacteria have genes to actually couple with other bacterial cells. And they form the sex pilus, which will connect the two cells together. Genetic information, either from the separate plasmid or from within the bacterial chromosome, are passed to the other cell. Next, we're going to get into transduction, but that'll be a separate lecture. All right, so just make sure you know the terminology and an overview of both processes.